We opened a new laboratory this April with the goal of developing drugs that improve the functional prognosis of patients with intractable neurological diseases such as stroke and dementia. Currently, as society ages, stroke and dementia have become major causes of death and disability. As these pathological conditions progress, neurons in the brain will die. But as we still do not have effective treatment for these diseases, they are typical examples of intractable diseases. In the brain, as tissue damage occurs, inflammation is triggered by immune responses followed by neuron repair, and patients are expected to recover their brain function. Until now, the intrinsic brain mechanism that triggers brain function recovery was unknown. But we have discovered that PLG2E, a lipid metabolizing enzyme, generates neuron repairing lipids such as G-homogamma-linoleic acid and 50-HTRA to induce PADI4, a pivotal transcription regulators for repair-associated gene expression in brain function recovery. Neuroinflammation is a bioregulatory process against brain injury. Glial cells and infiltrating immune cells such as macrophages and microglia are the first to respond to damage and produce inflammatory factors worsening patient outcome. It has been generally believed that functional recovery following CNS damage is actually decided on the repair of the neuron circuit, so we are focusing on the effect of immune and glial cells on neurons being repaired in mouse model of CNS damage. By utilizing our technique to isolate neurons from brain samples, we are conducting next-generation sequencing such as single-cell RNA sequences and ATAC sequences to clarify the molecular mechanisms underlying the neuron repair. From the single-cell RNA sequences, we are able to describe how each and all cell types in the brain respond to inflammation, and from ATAC sequences, we are able to deepen our understanding of the molecular mechanisms underlying the biological process in brain functional recovery. With these findings, our lab aims to introduce novel therapeutics harnessing the endogenous brain repair mechanisms to promote functional recovery from CNS diseases. Thank you very much for watching.